Keep in mind you have a homework assignment. It's your last homework assignment, which is due Thursday. Okay, so make sure you get that completed. And then we'll start lab at usual time, but this will also be your last lab, and then we'll be finished with that. So we're in the home stretch right now. Also, I want to give you a reminder, don't forget your exam, your final exam has been moved up. It'll be a week from today, okay? So make sure you're keeping an eye on Blackboard announcements as well as the syllabus amendment, okay? I mean the schedule amendment. All right, let's go ahead and get going here. Listen carefully. From chapter 10, we covered DNA replication, we covered gene expression, okay? Gene expression, that part of chapter 10 where we covered gene expression directly feeds into chapter 11. Okay, so you have to know your information for gene expression to understand chapter 11 and even lab 12 today for it to make sense. So if some of this, what I go over, is just completely foreign to you, go back, look at chapter 10 gene expression, study that real well, watch the videos I gave you, then go forward to chapter 11 into your lab 12. So gene regulation. What we're talking about in chapter 11 is how genes are controlled in regards to gene expression. There are various ways the expression of a gene is controlled. Now, what I'm showing you here on this figure is what we call linear DNA. Eukaryotic cells contain linear DNA that has open ends. Okay, prokaryotic DNA is circular in nature. There are no open ends. So what we're focusing on right now is eukaryotic DNA. But remember, up here it says nuclear DNA. Over here I'm showing you a nucleus, so you should know, okay, that we're talking about eukaryotes. What they found is that chromosomes, when DNA is in the form of a chromosomes, chromatin, or sister chromatids, they found that the DNA in the nucleus of eukaryotic cells, okay, is actually packaged together with proteins. So I'll go to this next figure. These proteins are called histones. So over here they're showing you a sister chromatid, or two chromosomes attached to the centromere. They take one of these chromatids and they start pulling on it. Now up to now we've talked about these are only made up of DNA. Chromosomes, chromatin, sister chromatids. But truth is they're made up of DNA and protein. It's just the star of the show is DNA. So as they pull up on this tightly coiled substance here, and they start pulling up on it, notice you have a blue color, but there's also a purple color underneath the blue color. So as they continue to pull on what eventually is the blue DNA here, double-stranded helix, notice these little beads start to fall out. What they're trying to show you, okay, is that for eukaryotic DNA, the DNA that forms that chromatin, chromosomes, or sister chromatids is actually packaged together with proteins called histones. And what they came to find out, if you start here, is that the DNA, as we go from our left to our right and down, what they came to find out is that the way DNA is packaged together with these proteins called histones is that you'll have a certain amount of DNA, a certain stretch of DNA, wrapped around a group of 8 histone proteins. We call these 8 histone proteins a histone octamer. So right here, you have DNA wrapped 2 times or wrapped twice around a histone octamer. So when you go from here back to our right, they unwrap the DNA and they release the 8 histone proteins to show you this is our histone octamer. In blue is our DNA wrapped around these purple histones. Now, what else they found is in these structures here where you have DNA wrapped around a histone octamer, down here they show you, we call these nucleosome. Okay, so everywhere you have DNA wrapped around a histone octamer, we call the entire structure a nucleosome. So here's one nucleosome, here's a second nucleosome, here's a third nucleosome. But if you look here between this second one I'm pointing at and this third one, there's a little stretch of DNA that is unwrapped. Okay, I didn't put it on here, but write it down. This unwrapped DNA that connects one nucleosome to the other, we call that short stretch of unwrapped DNA linker DNA. Okay. Initially, scientists thought because of the way DNA is packaged in eukaryotic cells, in the nucleus of eukaryotic cells, they initially thought that the only place you would find genetic information along a stretch of DNA is in that unwrapped region or that linker DNA in between the nucleosomes. But eventually, scientists discovered no, you can just as easily find, or there's just as much of a chance of finding genetic information somewhere along the stretch of DNA that forms part of a nucleosome. And they came to find out that one way gene expression is regulated, one way gene expression is controlled, is just by the way the DNA is packaged together into these nucleosomes, connected in each nucleosome connected by linker DNA. But eventually what scientists found out is that the gene expression, in other words, gene regulation, and what we mean by gene regulation is the control of gene expression. So that's what this means down here, everybody. The way we define gene regulation, the control of gene expression. What this figure is showing you here, notice this is a nucleus, this is the cytoplasm again. That's up to you to understand. You're looking at a eukaryotic cell, a piece of it. But what they're showing you here, what they're trying to show you, is that gene expression can be regulated anywhere along the lines of transcription, post-transcription, translation, post-translation. But notice something. Instead of showing you light switches to represent controlling gene expression by having it on or off, turning it on or off, instead they give you these little valves. Because the way it works for gene expression is gene expression can be regulated either by turning it on or turning it off at the level of transcription or translation. However, gene expression can also be regulated by slowly turning it up or slowly turning it down. In other words, increasing the rate of gene expression or decreasing it, but never fully shutting it off or never fully allowing it to reach a maximum peak. That's why they're showing you these valves. These valves, you got to imagine, be representative of the valves you have outside when you turn on the water, turn off the water. Depends how much you turn the valve, you can control the rate of water flow. Well, that's what they're trying to show you. Anywhere along the lines of gene expression can be regulated. It's not just turning it on or off, it's how on or how off, well, increasing and decreasing. So again, what this chapter 11 is going to focus on is not just gene regulation, the control of gene expression, but down here, ignore the B. If you recall, gene expression consists of two steps. Transcription is the first step. Translation is the second step. Chapter 11 focuses on the regulation of gene expression at the level of transcription. So going into chapter 11, you have to know the key parts of transcription, such as what is the enzyme in both prokaryotes and eukaryotes that catalyzes this first step called transcription. It's RNA polymerase. You have to know that RNA polymerase will not use both parent double strands as a template, but it will only use the reverse strand, only the reverse DNA strand as a template to synthesize RNA. And that RNA is not any RNA. We call it actually messenger RNA. But you also have to know going into this chapter, RNA polymerase cannot locate the gene in both eukaryotes or prokaryotes. It needs help. So it needs help from a DNA regulatory sequence called a promoter. This promoter does not code for the protein. It simply helps RNA polymerase not only locate the gene it needs to transcribe, but locate the beginning of the gene. Once RNA polymerase binds to the promoter, it slides over to the beginning of the gene, unwraps the double-stranded DNA, creating a transcription bubble, takes the reverse strand, and starts synthesizing RNA depicted in red. However, RNA polymerase not only needs help locating the gene and knowing where to begin transcription or synthesis of mRNA, it needs to know when to stop. So opposite from the promoter, on the other side of the gene or downstream, we have another DNA regulatory sequence, doesn't go for the protein. However, it does let RNA polymerase know when it has finished this first step, transcription. We call this the terminator sequence. Okay, so you have to keep these in mind as you go into this chapter because it's advanced information is basically what we're talking about. Well, in eukaryotes, what they came to find out is the rate at which RNA polymerase works. In other words, how active or inactive RNA polymerase is when performing transcription can be influenced or can be affected by proteins called transcription factors, which are depicted in purple here. RNA polymerase is in this brown color here. This green stretch here, that's our double-stranded DNA, but they colored it in solid green to let you know it's a promoter. This is our double-stranded DNA down here, but they colored it in to let you know this is a gene. But if you go back upstream, what we call upstream from the promoter, notice the DNA bends. But you can see they colored in some stretches of DNA called yellow. We'll talk about this in a second, enhancers. But they show you these purple guys binding to these yellow stretches of DNA, bending the DNA to where they come in contact with RNA polymerase. These purple guys are proteins called transcription factors. Transcription factors are proteins that influence the rate of activity of RNA polymerase. They can either increase the rate of RNA polymerase activity or help activate RNA polymerase, or they can slow down the rate of RNA polymerase activity, or they can just flat out stop or inhibit RNA polymerase from being active at all. So transcription factors, there's many different types. Initially, what we do is we break them into two groups depending on whether or not they activate or make RNA polymerase more active. Those transcription factors we simply call activators. The other group of transcription factors, the other group of these proteins called transcription factors, these are the ones that either decrease the rate of RNA polymerase activity or altogether stop or inhibit. We call these guys repressors. Okay? The figure down here, these purple guys here, they actually represent activators. Now, how can you figure this out using this figure? Well, the way these transcription factors are able to influence or affect RNA polymerase activity is by their ability to bind to stretches of DNA away from the promoter and the gene and the terminator sequence. In other words, these transcription factors bind to additional DNA regulatory sequences known as either enhancers or silencers. Okay? Transcription factors called activators, they either activate RNA polymerase or increase its rate of activity, and the way they do it is a group of these transcription factors called activators together will work as a group. Some of them will bind to stretches of DNA that they recognize as regulatory sequences called enhancers. So you have proteins binding to stretches of DNA regulatory sequences called enhancers. With the help of other activators, they will bend the DNA over to where they can reach over to RNA polymerase. And now basically what they do is tell RNA polymerase either to become active or increase the rate at which you're performing transcription. In other words, increase the rate of how fast and how many molecules of mRNA you're making. However, we could easily switch these labels all out. We could easily change this label from enhancer to silencer, and if we did that, then these purple guys would represent repressors, which work much the same way. That's why they're only showing you the activators in purple binding to DNA sequences called enhancers here. Because all you have to do is pull and plug in this group, except the repressors, which slow down the rate at which RNA is performing transcription, are altogether inhibited. Well, everybody, it's these transcription factors in our body, okay, in multicellular organisms, that allow our cells to differentiate, that allow certain cells to express genes that other cells can't, such as the insulin gene. A lot of people don't know, but when we talk about type 1 diabetes, okay, the only cells that are capable of turning on the insulin gene located in your pancreas are being killed off by your own body. So as of right now, type 1 is usually said to be an autoimmune disorder, although it's not conclusively proven. What people don't know, including many type 1 diabetics, is as those cells, the only cells that can activate that insulin gene and produce insulin, are being killed off, the rest of the cells that make up your body still contain that. They also contain that insulin gene, but they don't have the correct transcription factors to help them activate that gene. Okay, so this is something that a lot of people don't know. So therefore, we're going to transition to eukaryotic cells gene expression. In your notes, I break it up into 3 parts. All cells of our body that make up our body, let's say the human body, every cell of our body should contain the same genetic information. If you recall, sperm fertilizes the egg. Mom and dad both donate one set of 23 chromosomes. That fertilized egg transforms into a zygote, which is a unicellular diploid cell. Every cell of your body comes from that diploid cell. So whatever genetic information is contained in that zygote, it is passed on to every cell that forms your body. This is something you should be familiar with from your cancer gene lab and your paternity testing lab. It feeds into this. Therefore, every cell of your body should contain the same genetic information. That doesn't mean every cell of your body is capable of turning on the same genetic information. However, there's certain genetic information contained in every cell of your body that every cell of your body is capable of activating. Example, they're showing a pancreas cell, a cell of the eye, and a cell of your nervous system. And they're trying to show you here. Any gene that codes for an enzyme involved in glycolysis, well, you have to remember, what is glycolysis? It's the first step in cellular respiration. What is cellular respiration? It's a cellular process whereby cells produce energy in the form of ATP. Well, every cell of our body has to be able to perform this process or else they'll die. Therefore, We have genes that we call housekeeping genes. Housekeeping genes are genes that every cell of our body have to be able to express, such as genes that code for enzymes involved in glycolysis. Notice down here, any rectangle or pillar colored in means we have an active gene. So it doesn't matter that these cells don't look the same and don't behave the same. The bottom line is they all need to produce energy in the form of ATP. So any genetic information they contain in them, any gene they contain in them that codes for an enzyme involved in glycolysis, citric acid cycle, or oxidative phosphorylation, those cells have to express them and have to express them all the time. You will never have them turned off. These genes are always undergoing expression, and the only thing that will happen is maybe expression will be enhanced, increased, but that's it. So we call these housekeeping genes, such as genes that code for enzymes involved in cellular respiration, such as glycolysis. But jump down here to the insulin gene. There are genes that only specific cells can express because they're the only ones that have special types of transcription factors that help RNA polymerase activate these genes. Case in point, your pancreas is the only organ that contains cells that have the ability to activate the insulin gene. The catch is this: 99% of your pancreatic cells are associated with your digestive system. Okay, that leaves 1% of your pancreatic cells associated with your endocrine system. And out of that 1%, roughly half of those cells, called beta cells, are the only ones that can activate the insulin gene. But notice here, they're trying to show you cells of the eye, cells of the nervous system, they have the gene also. The problem is they don't have the necessary transcription factors to help RNA polymerase activate the gene. Only beta cells have the ability. So some genes are cell specific. However, that's what allows for cellular differentiation. When that unicellular diploid zygote begins to divide, it forms a multicellular diploid embryo. But eventually, those embryonic cells start to take on special traits, special characteristics. For somehow, some way, some of them are only able to activate genes associated with producing muscle or transforming into muscle cells. Other cells, for some reason, will activate genes that allows them to differentiate or turn into nerve cells. Other cells will be able to, for some reason, to activate specific genes that allows them to turn on blood or become blood cells. So it's this ability of certain embryonic cells to be able to activate cell-specific genes that allows for differentiation or cells developing into different types of cells, such as nerve cells, skin cells, and so on. But then we have our last group of genes. These are genes that are expressed only when needed. Best example of that is cell division. Okay. We've talked about this before. Proto-oncogenes. Proto-oncogenes code for growth factors that promote cell division. Well, specifically, proto-oncogenes code for growth factors known as cyclin CDKs, which they're showing you 3 different cyclin CDKs. These 2 are the same. Okay. You can see here it says G1, S phase, and mitotic cyclin CDKs. These are growth factors or proteins coded for by proto-oncogenes. These genes, these proto-oncogenes, are only activated when a cell needs to divide. Now, early on in life, especially once we have development of the embryo, and the embryo differentiates into a fetus, then the fetus develops into a newborn, an infant, and an offspring, these proto-oncogenes are constantly being activated. But as we reach adulthood, okay, some cells stop expressing these. Other cells will activate the proto-oncogenes, but only when they need to divide, such as epithelial cells of the skin. So some genes, cells of our body are capable of turning on, but they won't turn them on unless they need them. Okay, so you can break them up into 3 groups along that lines. But much of what is known about gene regulation in eukaryotes actually came from prokaryotes, such as the bacterium E. coli. They found that prokaryotes, such as the bacterium E. coli, their genetic information, it has evolved to the point to where it's, arranged in a very efficient manner. Okay, case in point, in bacteria such as E. coli, they discovered that sometimes you will find clusters of genes. In other words, you will find 3 or more genes coding for 3 or more active proteins and the genes are located one attached to the other. You will not see this structural arrangement at the level of DNA in eukaryotes. Again, you will not see this level of structural development in DNA in eukaryotes. You only see it in prokaryotes. Well, what geneticists or scientists eventually discovered is wherever you come across a cluster of genes in a bacterium such as E. coli, well, the reason why you find these genes clustered together side by side is because they all code for proteins that are involved in the same function. So it's more efficient, okay? It's more efficient if if a cell needs more than one type of protein to be involved in the same function, then instead of having each of these genes regulated by a separate promoter, it just makes more sense to put them all together and put them under the control of a single promoter. They came to call these clusters of genes regulated by one promoter operons. The most popular operon is called the lac operon. The lac comes from the milk sugar called lactose, the disaccharide called lactose. What they found is that this cluster of genes called the operon, or more specifically, this cluster of genes that they now refer to as the lac operon, okay? The lac operon consists of three genes, LAC-Z, LAC-Y, LAC-A. And what they found is each of these genes codes for an enzyme that's involved in the metabolism or the breakdown of lactose to use it as energy. They found that this E. coli's lac operon consists of three genes, each one right next to the other. The LAC-Z gene codes for an enzyme called beta-galactosidase. Its job is to break down lactose from a disaccharide to monosaccharide so it can be utilized as energy. The LAC-Y gene was found to code for an enzyme called permease. It helps E. coli take up more lactose once it detects there's lactose present so that beta-galactosidase can break it down. The LAC-A gene codes for a protein called transacetylase. Now, the function at one time was unknown, and I've left it unknown. They do know pretty much what the function is. We're not going over it because it's not worth it, okay? But it is involved in lactose metabolism. So you can see here, everything boxed in is part of a cluster of genes involved in the metabolism of lactose, and we call it the lac operon. The LAC-Z gene is right up against the LAC-Y gene, which is right up against the LAC-A gene. Codes for beta-galactosidase, codes for a protein enzyme called permease, codes for a protein enzyme called transacetylase. Notice right here is a terminator. If you go upstream, notice there's a promoter. However, in between the promoter and the first gene, there's a sequence that you don't see in eukaryotic cells called an operator. But this operator behaves much like a eukaryotic DNA regulatory sequence called a silencer. We'll talk about this in a sec. The operator serves as the off switch, preventing RNA polymerase from initiating or catalyzing transcription. Notice that's why it's positioned between the promoter and LAC-Z. The promoter still has the same function. Its job is to help RNA polymerase locate the genes, but now we have an additional player. Now, if you come upstream over here, I'm showing you a separate gene, the LAC-I gene. Please listen carefully. This gene is not part of a cluster of genes that form the lac operon. This is its own gene. Notice, promoter, LAC-I gene, terminator. The reason why I'm showing you this here is because the LAC-I gene is known as the lac repressor gene. Okay, the LAC-I gene codes for a protein called lac repressor. Okay, the lac repressor turns off or represses the lac operon by inhibiting RNA polymerase. So what they found is that this gene codes for a transcription factor in prokaryotes, such as E. coli, that functions as a repressor. And what will happen is from this gene, when it's activated and an expression occurs, we get a protein called the lac repressor, and that lac repressor recognizes and binds to the operator. And that's what they're showing you here. So up here they're showing you separate. Notice here also they're saying the operon. This is the lac operon gene, but outside of it, not part of the lac operon gene, they're showing you this represents the lac-I gene. It codes for a protein known as the lac repressor. This lac repressor protein is a transcription factor that recognizes and binds to the operator, which is part of the operon. And whenever it binds to the operator, which is located between the promoter and this cluster of genes, it interferes or inhibits RNA polymerase from attaching to the promoter. Now, some figures will show RNA polymerase still binding the promoter, but it doesn't matter. In either case, whenever the lac repressor binds to the operator, it inhibits RNA polymerase. Therefore, transcription is turned off. Okay, so we're going to go over this now, the way it works. So again, these are the working parts of the lac operon: a promoter, an operator, a cluster of genes known as LAC-Z, LAC-Y, LAC-A, and a terminator. However, we have a separate gene that is not part of the lac operon called the lac-I that we talk about because it codes for a protein called the lac repressor. Now, the way this works, notice here in parentheses, the way it works is that the lac operon, gene expression of the lac operon is usually turned off. Okay? The only way this will be reversed, the only way the lac operon will go from being turned off to turn on is if lactose is present. Well, lactose isn't always present. It's a nutrient. So whenever lactose is not present, what happens is the lac repressor has a shape that allows it to bind to the operator. Okay, when it binds to the operator, it physically inhibits RNA from polymerase from binding to the promoter. And then, as I said, even if RNA polymerase could bind to the promoter, the lac repressor, by binding to the operator, is still physically blocking RNA polymerase from sliding over and initiating transcription. However, what they discovered is that the lac repressor here actually has two binding sites, but they both cannot work at the same time. So again, what was discovered is the lac repressor has two binding sites, but they both do not work at the same time. When lactose is not present, there's a binding site on the lac repressor that complements the DNA that forms the operator, allowing the repressor to bind to the operator, inhibiting RNA polymerase from turning on the operon. It inhibits gene expression by interfering with RNA polymerase. However, when lactose is present, what happens is the other binding site on the lac repressor, okay, has a shape that complements the shape of this disaccharide or this milk sugar known as lactose. And what they found is even though the lac repressor has two binding sites, the binding site on the lac repressor that complements and binds to lactose, okay, it will work first whenever the operator and lactose are present. In other words, the lac repressor would rather bind to lactose than the operator if it has a choice. So when lactose is present, what happens is the lac repressor, instead of binding to the operator and inhibiting RNA polymerase, it prefers to use its other binding site to bind to lactose. But notice something. Remember we talked about a protein shape is directly related to its function? Well, when the lac repressor decides it wants to bind to lactose instead, it binds to lactose, but that causes the shape of the lac repressor to change to where the other binding site is lost. So when the lac repressor binds to lactose, it loses its other binding site, and therefore it can no longer bind to the operator. Now, RNA polymerase that has been hanging around waiting for the repressor to get out of its way, the repressor's out of its way, RNA polymerase now can bind to the promoter, slide over and locate the LAC-Z gene, and initiate transcription. However, the way it works is when RNA polymerase is able to initiate transcription now, it'll transcribe all three genes as one molecule of mRNA. But when this one molecule of mRNA is taken to the ribosome, along with the help of tRNA, is able to recognize that even though this is one molecule of mRNA, it contains genetic information on how to make three separate protein enzymes. So when translation occurs, it actually occurs 3 times, resulting in 3 separate protein enzymes. But the only way the lac operon will be turned on is if lactose is present, because the lac repressor prefers to bind to lactose, but that causes the lac repressor to change its shape to where it can no longer bind the operator. This in turn allows RNA polymerase to bind to the promoter of the lac operon, thus turning on gene expression. So what they found is for prokaryotes, which are considered the oldest known living organisms, they have evolved to where even at the level of DNA, even at the level of their genetic information, okay, it's arranged in a highly efficient manner to where somehow, some way, okay, at the level of DNA, the organism knows, hey, if I need beta-galactosidase, that protein enzyme, I know I'm going to need permease. I know I'm going to need transacetylase. I know I'm going to need all 3 enzymes. So let me save myself time. Let's just put them all together under the control of one polymerase. Because when I need one, I need all of them. This is the most efficient way. You don't see this arrangement in eukaryotic DNA. They've only discovered this arrangement in prokaryotic DNA. Okay, catch my breath here. Are there any questions? Okay, I'm going to stop the recording.